Hello, my wonderful and lovely humans. Today we are going to be taking some notes on DNA. So we have just finished talking about mitosis and all of the different steps that go into replicating brand new cells so that our body can heal itself and grow new cells and become bigger or, heal wounds, this, that, okay? And now we're going to talk about the DNA specifically, because we talked a lot about how the DNA acts differently, especially during all those steps of cell replication. So now we need to kind of understand DNA itself. So today's notes are going to be about DNA. Okay, so you guys should have a copy that kind of looks like this. Should have some blank spots. Okay. So make sure you guys are paying attention to the lecture so that way that you guys are able to copy down whatever it is that you need. Okay. So let me go ahead and flip my screen. All righty, so DNA structure notes. Let's get started. Okay, so DNA is actually, I know you guys have heard DNA a lot, right? But here's what it stands for. Deoxyribonucleic acid. So altogether, it's deoxyribonucleic acid. Deoxyribonucleic acid. So don't worry too much about this. I just want you to know what it's called. It's not necessarily something that you guys actually have to remember, but it is something that you do need to at least kind of know about. So yeah. Anyways, what I do want to point out here as we're looking at it, sorry, gotta grab my materials. Is that D, right? If we look here, is D. And then let's see, I need two more colors. Whoops, sorry. Okay, N is going to stand for right here, nucleic. And then that A right here is going to stand for acid. Okay. So deoxyribonucleic acid. Okay, but anyways, just to point that out. I think it's kind of neat. Alrighty, so deoxyribonucleic acid, aka DNA, is responsible for storing the genes, which is our genetic information that's going to code for our traits. And when we're talking about traits, we're talking about things that are characteristic of us. So like, for example, Miss Reyes has brown hair that grows out of her head. It's not actually blonde, believe it or not. My eyes are also brown, this and that. Yours might be blue or green or hazel. All those traits, okay? The things that make us different. So DNA is like the recipe book / instruction manual for how to build our traits and physical composition. And what I mean by composition is simply that, like, that's how it's built. That's how you are made up. So if you guys think of, I don't know, when you guys get Legos, a Lego set, right? Like you're building, I don't know, what's called the star thingy from Star Wars? I don't know. If you guys are building that, right, you have an instruction manual that tells you what Legos need to connect to each other and that stuff. Okay, so it's also going to be the building instructions / blueprints that are going to allow us to build living organisms. And when we say living organisms, guess what, buddy? That includes you and me and they and them and we and she and, everyone, all living beings, right? We're all living organisms. If you're listening to this, you are probably a living organism, unless somebody's like weirdly, like, I don't know, started playing, even the plants, but okay, anyways. All right, DNA is going to constantly be copied and replaced by the body in order to preserve our genes. And by preserve, we mean to keep it safe, to keep it consistent, to make sure that no mistakes are being introduced into our coding. So yeah. All right. Lastly, damaged DNA can range from unnoticeable changes to major issues / diseases and even death. So what I want to point out about damaged DNA is that we're going to go deeper and deeper into this topic with a future assignment because it is important to understand what DNA damage can result in. But it is important to know that overall, you can either have some unnoticeable changes or you can have some majorly life-threatening changes that happen as a result of DNA being damaged. Okay. All right. So now we can actually talk about the location where all of this DNA is actually residing and living and doing its thing. So DNA can be found inside what is called the nucleus of the cell. DNA that's in the nucleus is being protected and is also separated from the outside cytoplasm. And the cytoplasm, if you guys think back to what's going on in the cell, it's kind of like that jelly-like matrix stuff that's kind of holding everything in place, right? But here's the nucleus here in the middle. It is, if you guys look at this, right? So this is a cell, and it is encased in what is known as a cell membrane. We've talked about this before, right? And we've also talked about this really special word that's called 2 layers of selective permeability. Permeable security. So 2 layers of selected permeable security. So that cell membrane provides the first level of security, whereas the nucleus or the nuclear envelope, okay, so wait, cell membrane is like our first line of defense, okay? But then a second line of defense is called the nuclear envelope. And that's our second line of defense, because if you gotten past the first line of defense, now you are up to, the last one, right? So that would be this guy right here. So we're talking about the cell membrane out here. I know this is so bad. Oh my gosh, it's killing me. And then we're also talking about our nuclear envelope here. Okay. All right. So selectively permeable means that only certain things can get in or out of the cell. This is going to provide us with safety for our very valuable DNA. Because remember, DNA codes for everything we are, everything we ever will be, everything we will ever do, yada, yada, blah, blah, blah. Okay. All right. The nucleus is held in place by the cytoplasm. By the cytoplasm of the cell that surrounds it. So if you guys need to, you can pause this video so that you guys can go ahead and copy whatever you need to. Alrighty, moving on. I'm not going to flash you the whole thing because I want to actually make sure that you guys are, stopping the video and like watching the video, because realistically, this isn't helping you unless you're actually listening to everything. I'm just evil enough, remember? Okay, anyways, let's now talk about the structure of DNA. So what you guys need to know is that when you put 2 strands and you put a twisted ladder structure, and we'll talk about what this means here so it's not too confusing. When we put 2 strands together and a twisted ladder structure together, what we get is known as a double helix structure. So that is the structure that DNA kind of takes on, and it ends up being this like spirally staircase looking thing. So we're gonna talk a little bit more. So these two strands, each strand is going to be made up of a chain of nucleotides. Made up of a chain of nucleotides. And each nucleotide is going to be made up of 3 things. So the first one is going to be a 5-carbon sugar. All right. And that 5-carbon sugar is either, depending on what it is, so for DNA, it's deoxyribose, and for RNA, it's actually going to be ribose. And if you guys think back to way back when, we said whenever a word ends in "ose," it means sugar. So basically, this is how your body distinguishes between this stuff called DNA and this stuff called RNA. But both are really important in that DNA aspect, the coding aspect, the language of your body and how it is built. The other thing that you're also going to need is a phosphate molecule. Which, if I forgot to kind of point these out. Anyways, here's the 5-carbon sugar here. If you guys look at the 5 points, here's 1, here's 2, here's 3, here's 4, here's 5. That's what it's referring to with the 5-carbon sugar, because basically what's at each point here is a carbon molecule. So anyways, it's a 5-carbon sugar. We're also going to have this phosphate molecule, which is kind of connecting each piece here. You'll see how they fit together. And lastly, we're going to have this thing called a nitrogen base. And this nitrogen base can be one of 4 things. There's 4 different nitrogen bases that we can have. And we'll talk about this at the end here in a second. But there are 4 different nitrogen bases you guys can choose from. And that's what gives your body variation, right? It's going to rely on the order that some of these are being built in, but also a couple of other things that basically provide instructions to your body on how to build it. So when we start to chain them together, they're going to look like this. This is one strand of DNA. So once you put them together, as you guys can see, these phosphate molecules, they kind of fit nicely right into the 5-carbon sugar, and they make a chain. So both strands are going to form what is called a backbone. And if you think about your backbone, right, it's the thing that's in your spine that kind of provides you with some structure and supports. And that backbone that's being built here is going to do the same thing for the DNA. So if you guys need to, you can pause the video and copy whatever it is that you need to. All right, so we talked about the 2 strands portion of the DNA. Now we need to talk about the twisted ladder portion. So let's talk about it. So this twisted ladder, sorry, hold on. This twisted ladder, the backbone and, hold on, the backbone and the rungs of the ladder, so the steps, like these parts right here, okay? So you know how you step up the ladder? The rungs are going to make the DNA look like a ladder structure. And I know all of y'all have seen like a ladder or something of the sort, and that's pretty much what it looks like, right? Well, the thing is, when you start to twist that ladder, it ends up looking like this curly cube structure here, which I don't know if you guys have ever been in a house or a apartment or whatever that has a spiral staircase, but it looks very similar. So when condensed, all right, when condensed, the DNA is going to twist around itself, and it ends up looking like this twisted ladder structure. Yes, right, that twisted ladder. It's this twisted ladder structure right here. And it's going to ring itself, like completely condense itself tight like a towel. And it's going to make it to appear smaller, but it means that you're going to see it more visibly. You're going to actually be able to see it with your naked eye. So I'll show you guys here. I'm going to flip my screen over, and I need to go grab a towel here. but let me flip my screen because I want to show you guys what it actually kind of ends up looking like here. So basically, right, let's pretend this is our DNA. Can you see our DNA very well here? If you're looking head on, no, you cannot. But if I start to fold my DNA in on itself again and again and again, all right, now are you getting to see it better? Yes. And then I'm going to twist and 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 I'm going to twist. And I know you guys have wrung out like towels before, this or that, right? And when you twist and you twist and you twist and you twist and twist and twist and twist, right? Eventually it becomes teeny tiny. So we went from something that we could barely see to a very twisted up, balled up structure that we can now see very, very well. So just to remind you, this is what it looked like at the beginning. Okay. Oops, wrong side right here. Okay. So yeah, that's basically what happens. DNA is going to condense itself, and that's why we can see it becoming more visibly when we get to that prophase portion of mitosis. All right, back to the notes. Okay. All right. So the rungs / steps of the ladder are going to be made up of 2 connected DNA-specific nitrogen bases. Two DNA-specific nitrogen bases. Now, remember what I said up here? So remember that each nucleotide is made up of 5 carbon sugar, a phosphate molecule, a nitrogen base. Well, these nitrogen bases, I said that there was a variation, right? Well, there's 4 that it could possibly be. It can be A for adenine. It can be T for thymine. It can be C for cytosine, or it can be G for guanine. Okay, so basically what you guys need to understand about these is that A will only ever pair with T, and C will only ever pair with G. So A and T, they have these structures that are only going to be able to fit with each other. You guys are going to be able to play around with that when you guys get to the DNA lab portion of this, but I do have some set up here. So if you guys need to pause that video. All right, but here's what I want you guys to understand about those particular nitrogen bases. I'm going to tuck this off to the side of the screen because I want you guys to be able to see what's going on over here. So here I have a whole bunch of little DNA bits, right? So here's my phosphate molecule. All right. Here's my phosphate molecule. Here's my 5 carbon sugar. As you can see, when you guys start to build these together, you start to form a chain, right? Well, what's going to happen on the opposite side of that is we're also going to be building another chain, right? Sorry, I'm going to have to put it away. We're also going to be building another chain, but this chain actually, if you look at it, right, if it's going to fit together, it can't be pointing the same way. So it actually has to go the opposite way. So this is important because basically what you guys are going to find out is that there's 2 different directions that DNA will run in. And that's called from 5' to 3'. So back over here, if you guys look over here, right here, okay, at this little ladder structure right here. Sorry, I know I messed it up. If you guys are looking at this little ladder structure right here, you'll notice that there's a 3 over here and a 5 on the opposite side. So remember how these had to point in opposite directions in order for them to work together? Well, that's what we're talking about with 5' versus the 3' end. So DNA always runs 5' to 3'. So this DNA strand on the right side is going to be running up and down, or sorry, down and up to down. Over here on the other end, it's actually running from 5' to 3', so it's going from bottom to top. So these are actually running opposite each other, and this is what's known as anti-parallel. Anti-parallel. So they have an anti-parallel, which means that they run opposite directions. They have an anti-parallel structure. So here you go. You can see they are running opposite directions so that they might fit together, right? Okay. Well, anyways, let me go ahead and get rid of that because back to here. Okay, now I have to reset. So hold on a second. Let me zoom out here so you can see everything. So as you guys can see, my DNA molecules are starting to fit together pretty nicely. Well, actually, these aren't my these are my 5-carbon sugars and my phosphate molecules, right? Well, anyways, I'm going to add my nitrogen bases in here. So that way I might actually get to see what it looks like as a built structure. So this one right here is the nitrogen base A. And if we look back at our sheet, that was A for adenine, right? So A for adenine is going to connect right here. Okay, wait, hold on. Want to make sure this stays on screen. Okay. All right. Well, nitrogen base A is, if we look at the rest of these puzzle pieces, right? So here is C, here is T, here is G. There's pretty clearly only one thing that fits. It's not going to be C because as you can tell, yeah, nope. This isn't the puzzle piece that fits. But this, on the other hand, this one might. So oh, look at that. It fits like a puzzle piece, right? So now I have a nucleotide over here. Now I have a nucleotide here. And here they're able to pair between their nitrogen base structures, okay? So I'm going to continue doing that, but I want to now show you with a different nitrogen base. So we'll now use a nitrogen base G. And again, quick reminder, so that T1 over here, that was thymine. Adenine only connects with thymine. Cytosine and guanine are only going to connect with each other. And then again, that is due to the shape. So if you guys look at this, I should have cut that out, but listen, y'all know I was cutting for weeks, okay? Leave me alone. Got a little lazy with it. It's okay. It's okay. But anyways, I know when I'm shaky too. Always shaky. Okay. Anyways. So now we have nitrogen base C and nitrogen base G. And as you guys can see, they connect pretty well together. All right. But they would not be able to connect with each other if they were to cross sections or whatever. A's and C's cannot go together. G's and T's cannot go together. They have a specific fitting pair, and that is it. There is nothing to be done about it. It is simply how it works. So that is what you guys need to know. Hold on, let me flip my camera. So you guys are going to be responsible for starting to play a little bit with your DNA molecules. And you guys are going to get little sets of these cards themselves. You guys are going to have to identify certain structures that we talked about in these notes. And that's pretty much it. Okay. So anyways, that might be on a different day though. We'll see. We'll see what Miss Future Reyes wants you to do. All right. That's all I got for you guys. Have a great day. Take care. Be safe. Bye.